It's my distinct pleasure to introduce today's grand round speaker, Dr. Saeed Sheikh. We have known each other for many years because of our mutual scientific interests, and we used to serve on several review committees together. So, Dr. Sheikh received his medical degree in Pakistan and then proceeded to New York, where he received a Master of Arts degree in genetics and molecular biology. Subsequently, he received a PhD in cell and molecular biology from University of Montpellier in France. He has long been interested in research related to identification of cancer biomarkers and development of new therapeutic strategies. He has identified and characterized several novel genotoxic genes and proteins that are linked to cell death and survival signals in human malignancies. His research is well supported by grants from NIH, DoD, and other sources. And he did his postdoctoral work at the National Cancer Institute in Bethesda, following which he joined the faculty of SUNY Upstate Medical Center, Medical University, uh, where he is currently professor of pharmacology. Uh, he has been recipient of several honors, including Sinsheimer Fellow Award and election as Overseas Fellow of the Royal Society of Medicine in England. So today is the title of his talk today is Molecular Tumor Markers, Cellular Metabolism, and Tumorigenesis. Okay, go ahead, Dr. Sheikh.
Okay, well, thank you, Dr. Ahmed, for the kind introduction. And thank you for inviting me. So this is the title of my talk. And these are the learning objectives, because it's a grand round. And I do not have any financial conflict of interest to declare. So I do cancer research. So as we know, cancer is the second leading cause of death. More than 2 million new cases are diagnosed each year, and more than 500,000 deaths that occur due to cancer. So clearly, there's a significant morbidity and mortality associated with cancer. So as Dr. Ahmed mentioned, my lab interests are as follows: cell death and survival signaling in relation to cancer pathobiology. We are also developing targeted therapies and also developing tumor markers and investigating also the molecular basis of cancer responses to genotoxic and to plasmic reticulum and metabolic stresses. So we have over the years identified, cloned, and characterized several new genes, new at the time. So these are some of those I have listed here. So what I'm going to do is today I'm going to talk about two of these, CHTM1. It's a mitochondrial and cytosolic protein that is associated with the regulation of cellular metabolism, and also it's deregulated in human malignancies. And also CHCM1, which is also deregulated in human malignancies, and it's a DNA damage regulated mitochondrial protein. So CHTM1. So when we started out, nothing was known about this, only a hypothetical DNA was in the data banks listed as CHCHD5. So we named it as coil helix tumor and metabolism 1. Here's the nucleotide and amino acid sequence of this gene. And here's the genomic organization. It has 4 exons, and the open reading frame resides in the open in these 4 exons. And it has these coil helix domains, 2 of these. So there are cysteine residues that facilitate the formation of these coil helix domains. And this contains 110 amino acids, and molecular mass is approximately 13 kilodalton. So once you have a new protein that gene you wanted to characterize, so first thing you do is that you wanted to have an antibodies against it. So what we did was we expressed that in bacteria, purified it as shown here, and then generated polyclonal antibodies in rabbits. So, and we tested, and the antibodies were working very well. So then we wanted to see where it resides inside cells. We perform immunostaining. And as you can see here, using these breast cancer cells, as you can see here, it's a green punctated distribution was noted. That indicated to us that it was residing in some cellular compartment. And then we also stained these cells with mitotracker, which is mitochondrial specific dye. And when we compared, the distribution pattern was similar, indicating us that this was in mitochondria. Then we did biochemical analysis. We prepared these cytosolic as well as mitochondrial fractions. And we performed Western blotting using the antibody that we generated. And as you can see here, so we found it to be present in cytosol as well as in mitochondria. So as a positive control, we use probably same blot with the CHCHD4, which is mitochondrial protein. And we found that to be in mitochondria, but not in cytosol, indicating that the fractions were clean. Tubulin is present only in cytosol, and as expected, we found it to be only in cytosol, but not in mitochondria. So these results confirm that this protein was in both cytosol as well as in mitochondria. So what was the function? So that's what we then started to investigate. So what we did was we generated a knockdown using breast cancer cells, as is shown here. We generated 3 knockdown clones. These are scramble, and these are the knockdown, these are Western, indicating that the levels of CHTM1 were significantly lower, which means that the knockdown was working. And because this was in mitochondria also, so we sought to investigate its effect on mitochondrial function. We look at the oxygen consumption rate, and we found that that rate was reduced in the knockdown cells, and ATP generation was also reduced, indicating to us that this was affecting, its deficiency was affecting mitochondrial function. So then we wanted to continue on to investigate its function. So we asked this question, okay, how its deficiency will affect cell growth? And strangely, we found that when we knock it down in cells, so the growth was not affected. So we played around some conditions, and finally we found that when we deprive the cells with glucose and glutamine, the nutrients, so their growth was affected. And this is a representative of photomicrograph of scramble cells grown in these nutrient-deprived conditions. And this is a clone that is deficient in CHTM1. These are photomicrographs showing that the growth was compromised, that they grew poorly in nutrient-deficient conditions. Down below here, we are showing the crystal violet staining of the cells. These are scrambles, and these are 2 knockdown clones. And as you can see that the knockdown grew poorly in nutrient deficient conditions. So those were breast cancer cells. We also investigated that in lung cancer cells, and we used 2 different lung cancer cell lines, A549 and H460, knocked it down, shown here, the Western blot, that knockdown was very good. And we found that similar results, that their growth was also affected under nutrient-deprived conditions. And these were the quantitative results. And here we showed you qualitative results. As you can see that the cells that are deficient in CHTM1, They grew poorly. These are crystal violet staining. These are scrambled cells, and these are phase contrast photomicrographs. And as you can see, their cells are growing poorly in nutrient-deprived conditions, those that were deficient in CHTM1. And that was the case for both lung cancer cell lines. So as we know that glucose is metabolized through glycolysis, is converted eventually to pyruvate, and under aerobic condition, it feeds into TCA cycle. Okay, so now glutamine is a non-essential amino acid, and it is converted to glutamate, and eventually glutamate feeds into alpha-ketoglutarate, which is one of the intermediates of TCA cycle. Now, glutamine and glucose, these are very important nutrients, and their deficiency leads to stress. We call it metabolic stress. And glutamine, although non-essential, it can be conditionally essential, particularly under the conditions of cellular stress. So, so then we said that, okay, well, let's look at those clinically relevant drugs that are known to induce metabolic stress, and we found metformin. Now, metformin is an anti-diabetic drug. It's a fairly safe drug has been used for type 2 diabetes for many years. And it is also listed as one of the essential medicines by WHO. And it has been investigated in clinical trials against various malignancies. And like I said, it's known to induce metabolic stress. So we then use this also in our study. And we found that indeed, this also induced metabolic stress, and these cells, lung cancer cells that we used here, that were deficient in CHTM1 also grew poorly. So metabolic stress that was induced by these nutrient deprivation, as well as the clinically relevant drug, also was tested. Okay, so we investigated what was the mechanism, how these cells were poorly growing, and we found that they were undergoing cell death. Interestingly, what we found was that their cell death was occurring in a caspase-independent manner, as is shown here. It's because we use that as ZVAD, which is a pan-caspase inhibitor, and we found that it didn't interfere with their ability to inhibit cell death. So then we wanted to further investigate what was the mechanistic basis of its deficiency that was making them grow poorly under metabolic stress conditions. So because there was caspase-independent death that was associated, so we directed our attentions toward AIF1. Now, what is AIF1? It's a mitochondrial oxidoreductase. So it translocates from mitochondria to nucleus, and it is known to be involved in inducing caspase-independent cell death. Now, what it does in nucleus is that it induces DNA fragmentation and also chromatin condensation, the hallmark of cell death, but that occurs in a caspase-independent manner. So therefore, we looked at AIF1. So what we did was we had these cells, as I mentioned, which were knocked down, and obviously the scramble control. So what we did was we stained those with antibodies against AIF1. So what we are showing you here is the immunostaining detecting AIF1 in cells. So these are scrambled cells grown under regular conditions and nutrient-deprived conditions. So this, if this is present in mitochondria, so AIF1 specific staining will show like punctate distribution, as is shown here. And this is mitotracker, which is a mitochondrial specific dye. So the distribution is similar, as is shown here. So it's a punctate distribution. But once it gets out of mitochondria, AIF1. So the staining becomes diffuse. Okay, so here's what we found in the knockdown cells, CHTM1 knockdown cells. These are grown in regular conditions. The distribution pattern that you can see is punctated, indicating it to be in mitochondria. However, when they are deprived from nutrients, that is glucose and glutamine, the distribution becomes cytosolic, diffuse, as well as nuclear specific, as you can see that, right? So then what we did was quantitative these, and what we found was that unlike scrambled cells and cells that were grown in a regular condition, the mitochondrial distribution was decreased, nuclear distribution was increased, and cytosolic distribution was also increased. So we found similar results when we used metformin, which we mentioned is a clinically relevant drug. And as is shown here, again, that mitochondrial distribution was reduced, nuclear distribution was increased, and so was cytosolic distribution. So these indicated to us that this deficiency was affecting AIF1 translocation. And because CHTM1 and AIF1 both are in mitochondria, so the next question we asked was whether they interact with each other. So what we did was we transfected the cells exogenously in these HeLa cells with this CHTM1 vector and stained that with the anti-HE antibody, as is shown here, red color. This is exogenally expressed CHTM1. So this is endogenous AIF1 that we stained with the AIF1 specific antibody, and we found them to be present in the same compartments, that is co-localized. Now, two proteins, if they co-localize, that does not mean that they interact with each other, so we needed to do more. So we did further biochemical analysis. We performed this pull-down assay as well as immunoprecipitation, when we found that the exogenous CHTM1 was able to pull down endogenous AIF1 with it. And also immunoprecipitation, when we performed that, and we found that endogenous CHTM1 was able to precipitate endogenous AIF1 as well. So these results indicated to us that, yes, these proteins not only co-localize, but also interact with each other. Okay, so results so far indicate that CHTM1 regulates cancer cell sensitivity to metabolic stress, and AIF1 and CHTM1 interact with each other. And then it also regulates its translocation from mitochondria to cytosol, particularly under metabolic stress conditions. So because we're talking about stress, we knew that P38 is a well-known stress kinase. It's a mitogen-activated protein kinase. So we asked this question whether P38 kinase would be playing a role. So as you know, that P38 can be activated in response to various types of stresses, as shown here, and then it can activate its downstream kinases like this MAP kinase, activated kinase 2. And through this, it can, P38 can phosphorylate and modulate the function of various downstream substrates. And one of those is heat shock protein 27, which gets phosphorylated, and it is implicated in playing a role in anti-proliferation as well as in cell death. Although its role is dual. It has been implicated also in playing opposite role under certain circumstances, but in the case of cancer, particularly in response to stress, it has been shown to play an anti-proliferative role. So then we investigated that effect. So here what we are showing you is that these are Western blot analyses. What we did was we had these scrambled and knockdown CHTM1 knockdown cells, and we subjected those to metabolic stress through a nutrient deprivation. And as you can see, these are the cells grown under regular conditions, and these are the cells grown under nutrient-deprived condition, that is metabolic stress-inducing condition. And we analyze the phosphorylated version of P38, that is activated version of P38. And as you can see, under stress conditions, P38 phosphorylation increases, indicating it's being active. It's more so in knockdown cells compared to scrambled cells. And this is the total P38, which is change, indicating that only phosphorylation was enhanced. And then we look at the downstream substrate, which is heat shock protein 27, and we found that to be phosphorylated, indicating that that was being modulated and that P38 kinase was functionally active, and that cleaved PARP was also enhanced, indicating that that was translating into cell death also. So we then also use this P38 inhibitor to see if we can inhibit these effects. And indeed, we found that when we use P38 inhibitor here, so we can block that, inhibit that its effect on downstream substrates, that is heat shock protein 27 phosphorylation and also PARP cleavage. Okay, so these are qualitative results we're showing you here. So these are the lung cancer cells. These are untreated and these are nutrient deprived. These are scrambled, and these are CHTM knockdown. And as you can see, that when they are nutrient deprived, they grow poorly. But when we add this P38 kinase inhibitor, we can reverse that. So these are phase contrast photomicrographs, and this is staining, crystal violet staining, indicating that these are cells growing poorly. But here, when we add P38 kinase inhibitor, we can reverse it. Okay, so these results indicated that the inhibition of P38 activation is one of the mechanisms through which CHTM1 is promoting cell survival under metabolic stress conditions. So this led us to conclude that the results so far that I've presented. So we have this hypothetical model. We think that under metabolic stress, P38 kinase, which is known to be a stress kinase, it gets activated, and then it affects the AIF1 that is present in mitochondria, and its subcellular distribution such that it translocates from mitochondria, comes into cytosol, goes to nucleus, and mediate caspase-independent cell death. And CHTM1 tends to inhibit that here, and it also sequesters AIF1 in mitochondria and AIF1 in mitochondria and prevents its translocation from mitochondria into cytosol and then into nucleus and ultimately caspase-independent cell death. So moving on to further, we found that it was deregulated in human malignancies. So these are some of the results that I have. I'm going to show you. So we analyzed its regulation in various malignancies, and these are representative results for breast cancer. So we are showing you here the Western blot analysis performed on 6 breast cancer patients. What we did was we took normal tissue and matching tumor tissue, normal and tumor from the same patient. So this way we analyzed 6 patients, and N stands for normal, T for tumor. And as you can see that the tumors are showing increased levels compared to their matching normal tissue. Down below here, we are showing you the loading control, indicating that changes that we are noting is not due to loading differences. And we also performed immunohistochemistry staining, and we noted that it's also the levels were increased, and these are representative results on 2 patients. This is just normal tissue, and that's the tumor tissue. As you can see, the brown staining is enhanced, that is CHTM1 specific staining. So then here we summarize the results that we performed on 71 patients, and 61 samples out of 71 showed increased levels of CHTM1 that translates to about 86%. We also found that its levels were increased in colon cancer patients. Again, these are 6 patient samples we're showing as representative results. As you can see, the levels are increased in tumors compared to matching normal tissue. Down below here is loading control. And these are representative immunohistochemical stainings. This is normal, this is tumor, and as you can see that CHTM1 staining is enhanced in tumors compared to normal tissue. So overall, we analyzed 97 patients, and we found that out of 97, 79 patients samples showed increased expression of CHTM1, and that translates to about 81%. So in the case of lung cancer, these are representative results for lung cancer patients. Again, we found that its levels were also increased in lung cancer patient compared to matching normal tissue. Again, N stands for normal, T for tumor. As you can see that the levels are enhanced in the tumors because it's a Western blot. We probe the same plot with SAM50. Now, SAM50 is a mitochondrial protein. So the question that we were faced with that, okay, well, its levels are increased. It's a mitochondrial protein. So a question can be asked, yeah, its levels are increased because in tumors, it could be that mitochondria number were increased. So to rule in or rule out that possibility, we probe the same plot with SAM50. And as you can see that SAM50 levels are not regulated the same way as CHTM1 levels are, further confirming to us that it was levels increased was not because increased in total levels of total numbers of mitochondria, but rather specific to CHTM1. And down below here is loading control, indicating that the increased level of CHTM1 was not because of differences in loading. So we also perform immunohistochemical staining, and these are representative results from 3 different patients, and these are normal, and these are tumor specimens. And as you can see, its staining is significantly enhanced in tumors compared to matching normal tissues. Okay, so overall, we had 64 patients of lung cancer. So out of 64, we found 42 samples showing increased levels of CHTM1. So that translated to more than 65%. So we did a stratification also, and we found that we had 12 samples that were squamous cell carcinoma and 52 were adenocarcinoma. So out of 12 squamous cell carcinoma, 6 showed increased levels, and that translated to 50%. And in the case of adenocarcinoma, 36 out of 52 showed increased, that was like 70%. So overall, more than 65% specimens of lung cancer showed increased levels of CHTM1. So to summarize this part, CHTM1 is a novel modulator of cellular response to metabolic stress. So how does it function? So that is a key question that we're still investigating. But we think that early during cancer formation, cancer cells they have a limited blood supply. That's they're undergoing new angiogenesis. So under those conditions, there are less nutrients available for them. So we feel that increased levels of CHTM1 under those conditions confer a growth advantage to those cells, so they can survive and grow, the tumor-specific cells. And of course, it may not mediate effect alone, but in concert with other markers. And we also feel that it's pharmacologic or genetic targeting of this can be an important approach to treat malignancies. And also because we used a clinically relevant drug, metformin, that induces metabolic stress. We feel that drugs that induce metabolic stress, so this can affect the tumor cell sensitivity against those drugs also. So overall, we feel that cancer cell sensitivity can be enhanced if this is antagonized, CHTM1 is antagonized. Okay, so now I'm going to move to the second gene that we characterized, CHCM1. Again, at the time we were characterizing this, nothing was known about it except that there was this cDNA in the database annotated as CHCHDA6. So we named it coil helix cristae morphology 1. Then here we are showing you the amino acid sequence, and this contains DUF737 domain and this coil helix domain also. So this has 235 amino acids, and its predicted molecular mass is about 27 kilodalton. So what we found was that we analyzed its expression at the mRNA levels as well as at the protein levels. And we found that at the mRNA level, its expression was significantly downregulated when we treated the cancer cells with these anti-cancer drugs. We did the same thing that we did for CHTM1. We obviously generated antibodies, and we then analyzed its expression also at the protein levels. So these are representative results, Western blots, showing you the levels of CHTM1 in 4 different breast cancer cell lines. And as you can see, and these cells were treated with anti-cancer drugs, etoposide as well as adriamycin. And both are well-known anti-cancer drugs. And as you can see, then when the cells were treated with these drugs, etoposide or adriamycin, its levels are significantly reduced. So we found this to be regulated in response to these drugs. And interestingly, we found that it was specifically regulated in response to the drugs that were inducing DNA damage, that is genotoxic stress. So we had the antibodies that we generated. Then we obviously wanted to ask the question, where it's residing inside the cells. We performed immunostaining. So these are representative results. And as you can see, that this also showed a punctated distribution. And that was similar to mitochondrial distribution of mitotracker, which is a mitochondrial specific dye. So these results indicated to us that, yes, it looks like this is also in mitochondria. Then we performed biochemical analysis, and we found that, yes, it was also in mitochondria. So what we are showing you here, these are Western blots. What we did was we did fractionation. We prepared cytosolic as well as mitochondrial fractions. And as you can see that this CHTM1 is specifically in mitochondrial fraction. As a positive control, we probed the same blots with TIM23, which is known to be mitochondrial protein, and indeed we found that to be present in the mitochondrial fractions, indicating that our fractions were clean. Okay, so we characterize this further, which I will not go into too much detail because we don't have time. And my focus today is different. So we found this to be a subunit of mitochondrial contact site and cristae organization system, which is shown here. And this resides right here. So we found that this was involved in keeping the integrity of mitochondrial cristae. And that is why we give it this name, CHCM1. So what I wanted to show you today was this its deregulation in human cancers. So what we did was we analyzed its expression using cancer profiling array at the mRNA level. What we had was a normal as well as tumor-specific mRNAs from a large number of malignancies, as listed here: breast, ovary, colon, stomach, and so on and so forth. And we analyzed its expression, and what we found was that its expression was increased in breast, colon, lung, testicular, thyroid, and skin cancer. In the case of skin, it was melanoma specific and not other types of dermatological malignancies. So because the levels were increased at the mRNA, so we wanted to ask whether regulation was also occurring at the protein level. So these are the representative Western blots. What we did was we took samples from patients. These are colon cancer patient samples. And we are analyzing these at the protein level. But we are showing you here that CHCM1 levels were increased. These are representative results from 6 patients. So 3 out of 6 patients showing increased levels in tumors compared to matching normal tissues. In the case of breast, we found that these are representative results for 4 patients, and we found that its levels were also increased in the tumors compared to matching normal tissue. Down below here is the loading control. And we also analyzed lung cancer patients, 6 representative patient samples. Again, its levels are increased in tumors compared to matching normal tissues. So overall, we analyzed 17 breast cancer patients, and we found 9 showing increased levels of CHCM1. That translates to 53%. In the case of colon, we had 30 patients, and we found 15 out of 30 showed increased levels of CHCM1. That translates to 50%. In the case of lung, we had 27 patients, and we found 18 out of 27. That translates to 67%, showing increased levels of this CHCM1. Okay, so as I mentioned, we found that its levels were also increased at the mRNA level in melanoma. So then we also wanted to see its expression in melanoma at the protein level. So we got 26 patient samples, melanoma patient samples, and we also had 16 samples from normal skin. So we performed immunostaining, and these are the representative results. So what we are doing is we are showing you the immunostaining. There's a pink color. So what we are showing is that CHCM1 expression in melanoma and normal skin section is showing this in a melanoma. It's varying, I think I should say that, which is pink to red staining. And it's with strong to moderate to weak in the case of melanoma specimen. So these are, excuse me. So varying CHCM1 specific staining is shown here from red to pink. It is a strong to moderate. Now in the normal skin here, most of the superficial cells in the epithelial layer are weakly positive, as is shown here. And the basal layer also shows a weak staining with melanocytes shown here in with arrows. Excuse me. So this is again normal skins, and when we're showing you melanoma specific staining pointed by arrows, and we found that they were not positive. So here we have summarized these results. In the case of melanoma, we had 26 cases. 5 out of 26 showed increased, staining for CHM specific staining that translates to 19%. And 21 out of 26 showed increased staining that is moderate to strong staining that translates to more than 80%. In the case of normal melanocytes, we did not detect its staining. So melanoma, it's a very aggressive malignancy, and it arises in melanocytes. And there are different types of melanomas, such as cutaneous, acral, mucosal, uveal types. Cutaneous is most common. And BRAF mutation is one of the common hallmark of large number of these cutaneous melanomas. What is BRAF? BRAF is a serine threonine kinase, and it is mutated. The mutation occurs at valine residues. So, valine 600 is changed to glutamic acid. And that's a very common mutation that occurs in the activation domain. And that leads to constitutive activation of this kinase. Valine 600 to lysine is the second most common mutation. So currently we manage metastatic melanoma, as you know, through these BRAF inhibitors, such as vemurafenib and abrafenib, and also MEK inhibitors, trametinib and cobimetinib. So what's the mechanism of the actions of these BRAF inhibitors? So this is the pathway, BRAF pathway. Under normal condition in normal cells, this pathway is not always active. It is active only in response to mitogens when the cells need this pathway to be activated. So then it sends this proliferation and survival signals. But in cancers, so in which this BRAF is mutated, what happens is that this BRAF becomes constitutively active, and it constantly sends these proliferative and survival signals. So the BRAF inhibitors, they act at this level and inhibits its function, and thereby prevents this transduction of proliferative and survival signals. So these MEK inhibitors, they act at this level. So vemurafenib and dabrafenib, they act at this level. Trametinib and cobimetinib, they act at this level, that is MEK, and they prevent the transduction of proliferative signals that are emanating from this mutant BRAF, and thus inhibit the proliferative and survival signals. So that's the mechanism. So next we ask this question, that what will happen to these cells if we knock down the melanoma cells that harbor BRAF mutation? If we knock down CHCM1 and we treat them with this BRAF inhibitors. So that's what we did. These are A375 melanoma cells. They harbor BRAF mutation. And these are scrambled cells treated with vemurafenib, BRAF inhibitor. And it inhibited their growth, but not as much. But when we knocked down CHCM1, the growth was significantly inhibited. So these are quantitative results we are showing you in two different melanoma cell lines, A375 and UACC. Both of these, they harbor these BRAF mutations, and we found that the proliferation was significantly further inhibited when we took out CHCM1. So to summarize this part, CHCM1 is present predominantly in mitochondria, and the results I didn't show you, which were published, that it associates with inner membrane, and it bridges the interaction with outer and inner membrane, and its expression is significantly downregulated by anti-cancer drugs, particularly those that induce DNA damage. And its expression is deregulated in different malignancies, and its deficiency further enhances BRAF inhibitors' effect on BRAF mutant melanoma cells. So therefore, we feel that this is an important target. That is, CHCM1 can be therapeutically targeted. It can be antagonized to enhance the effects of anti-cancer drugs. And also, it has the potential to be an important tumor marker. Okay. So in the end, I'd like to acknowledge these are some of the people who did some of this work over the years. They've been with us. And the collaborators, my pathologist collaborators, Dr. Chris Carters, Steve Landers, and Dr. Sue. So I will stop here and will happy to answer any questions that you'll have. Thank you.
Thank you very much. It was a great talk. I invite questions. Let me start off. Sorry, go ahead.
I have a quick question, Khalil.
Yeah.
So the first molecule you talked about, CHTM1, and you were discussing how it could be a combinatorial approach with metformin. Have you looked at any other cancer drugs that affect other pathways other than metabolism?
Yeah, the results we didn't present here. So we have looked at, and currently actually we're investigating another drug. It's actually clinically relevant also. It's a CB839. So it we can say that it's a glutamine inhibitor. So that's currently in clinical trials. So we are looking at that, and we are finding similar results, although that work is currently in progress.
Okay, thank you.
Yes.
Anyone else has a question? I just have a question from my own information. I was wondering if CHGM1 or CHCM1 have any effect on the mitochondrial calcium signaling?
That's an interesting question, a very important question. We have not investigated that aspect yet. I would like to do that.
Yeah, because it's calcium sort of is the terminal cell death inducer, so I'm always interested in
right. So there are actually various other approaches one can take. In the case of calcium, obviously there is a crosstalk between the ER and mitochondria, because as you know that ER is important in context to calcium, but there is this crosstalk between ER and mitochondria. And then that happens also through calcium signaling.
Thank you.
Yeah.
Anyone else has a question? Guess it was a very clear-cut talk, so everyone has understood everything.
Well, let's close for the day. Thank you, everyone, for joining us. We do have a faculty APCP meeting at 9 o'clock, so timing is perfect. Thank you, Dr. Sheikh, Dr. Ahmed, and Dr. Trembley. This is our last grand rounds for the year. We have summer off. Thank you all for joining us. Have a good day.
Thank you. Hey, bye now.
Thank you, everyone. Appreciate it. Bye-bye.
Yeah.